Hello, my name is Leonor Leandro, and I'm a professor of plant pathology at Iowa State University. I conduct research on fungal pathogens of soybean, and today I'm going to be talking to you about some more recent research that I'm conducting looking at the potential for using plant essential oils for management of soybean fungal pathogens. So what I'm going to talk about today is asking the question, what are plant essential oils and what are their desirable properties? What is their potential as biopesticides? And then I'm going to provide an update on research that I'm conducting here at ISU in collaboration with other colleagues, and then some kind of key points about future research directions and potential implications of using these compounds. So what are plant essential oils? Many of you probably have heard about this term, have seen them in the context of health or therapeutic massage or some other aromatic components of these essential oils. But basically what they are concentrated mixtures of volatile aromatic compounds. And these are produced by plants as secondary metabolites. These mixtures have a liquid form, and so they look like an oil, and they can be extracted from different plant parts, from seeds to roots to leaves to bark. One key characteristic of these plant essential oils is that they are, again, volatile, so that's why they have an aroma, we can smell them, but they include a variety of compounds, including terpenes, phenols, alcohols, aldehydes, and several other compounds that are in a mixture from these different plant parts. What are the properties of these plant essential oils? They are well known for having very potent antimicrobial properties. They can be antibacterial, they can be antifungal. They're also well known for their insecticidal and also repellent activity. They can even be herbicidal, so they can have been explored as a potential for using as an herbicide. Because of these properties that they have, they are used in a variety of ways. They can be used for nutritional and flavoring applications. They are used in some medicinal applications, in cosmetics, also for food preservation. And a bit more recently, there's been really growing interest in using these plant essential oils as biopesticides. Post-harvest is probably the application that they're most commonly used. So after harvest, can we apply these plant essential oils and prevent rots from developing? But they're also being used as foliar sprays to combat diseases and also pests. There's interest also in these seed treatments. And you see there on the slide some images of some of the commercial available products that are out there. So why is this increasing interest in using plant essential oils as biopesticides? Well, we've all heard about the environmental concerns about synthetic pesticides. So you may have residues of these pesticides in water and in plants. There's pathogen resistance that can develop if these pesticides have a single mode of action or certain modes of action that are similar to what other pesticides use, so the pathogens can develop resistance. Many of them are also phytotoxic. And of course, then there's the public health concerns about applicator and exposure when these things are applied. So there's constantly a search for more sustainable approaches to control plant diseases and pests. What the essential oils bring to us is, one, they are a natural product extracted from plants. They are biodegradable, so they don't last very long in the environment. They also, because they have all those different compounds in those mixtures, they have different modes of action. And so they target different parts of the pathogen, and so the development of pathogen resistance is less likely. Some of them are also thought to induce plant resistance, so they can trigger sort of an immune response in the plant, making them more resistant to disease. And one of the goals of using them as biopesticides is maybe we can reduce the reliance on synthetic pesticides. And I'll talk about later that this could be in the form of using it instead of some synthetic pesticides, but also could be using in combination with synthetic pesticides, so that both modes of action are there. Maybe you could reduce a little bit the dose of the pesticide. The other benefit is that they are generally regarded as safe. There's an acronym GRAS that refers to these plant essential oils. They're used in food. And so the regulations and to be able to market them are not quite as strict as at least yet. And then the graph there shows that really there's been a gradual increase in market trends in wanting these more sustainable products, biopesticides, and so that's also pushing the interest in these products. Okay, so starting to talk about the research that I'm conducting in my lab. This has only been a couple of years when I started working on this, so it's a little bit early to have a lot of applications yet for growers. But what I want to share with you is that I have in my lab been screening different plant essential oils for activity against soybean fungal pathogens, specifically soilborne fungal pathogens, which is my area of expertise. So we're working on 4 different pathogens: Fusarium frugiliforme, I'll refer to it as FV, that causes sudden death syndrome; Macrophomina phaseolina, that causes charcoal rot; Pythium sylvaticum, which is a really important pathogen causing damping off on seedlings; and then white mold or Sclerotinia sclerotium that causes white mold. So these are 4 pathogens that we're targeting to screen these essential oils. I'd like to show this slide here because it's a very visual representation of the kind of impact these essential oils can have. Sometimes there's a little skepticism, well, they're plant essential oils, are they really effective? And what you're seeing this in this picture here is the first screen we did with lemongrass oil, essential oil, and its effect on the growth in petri dishes of Fusarium frugiliforme, so the SDS pathogen. And so what we do in our in vitro assays is we add the plant essential oils at different concentrations to the agar, the medium, and then we grow the fungus on those plates. And so at the left side of the slide, you see 0%. That means that's just control. That's just agar, the regular agar we use to grow the fungus. And the colony you see there is the fungal growth. I believe this is after 10 days. And then as you go towards the right, you see increasing concentrations of the plant essential oil, the lemongrass. So we have 0.03% by volume, 0.04, so forth until 0.08. And these concentrations correspond to 300 to 800 parts per million. So these are very small doses that we have added to the agar. And what you can see is even at 300 parts per million, 400, the growth of the pathogen has been reduced, but 500 parts per million and greater, you have absolutely no growth of this fungus. So this was very exciting because it's one of the first screens we did, and we were seeing how effective this oil is at suppressing Fusarium frugiliforme. So the research we're conducting here in my lab is then it started to expand into looking at other essential oils, ones that are known in the literature to have antifungal activity. Others have been recommended by colleagues who have worked on these pathogens. So you see a list here. We selected 16 essential oils that we have screened, have been screening for activity against those 4 pathogens. So again, we use potato dextrose agar. We use then now for all these screens, we only do the 300 parts per million and 500 parts per million because lemongrass already was effective at 500. So we wanted to see something that was as good or better than the lemongrass. And the tween there is important because it's an emulsifier. So essential oils are not easily emulsified in water, so we need to use the tween 80 to help them emulsify and distribute homogeneously in the agar. So our controls have the tween and all the treatments have the tween. Okay, so I'm going to start showing you some of the results we have had. The first set of slides is on Fusarium frugiliforme, so the SDS pathogen. And what you're seeing at the bottom on those petri dishes are the selection of, out of the 16 oils, 5 of the oils that have most strong inhibition of Fusarium frugiliforme. So cassia oil. Cassia is related to cinnamon, and it has been shown to be as effective or more effective than cinnamon bark. in suppressing growth. So you can see the little dot in the middle of those plates is the little plug of where we introduce the pathogen. So there's absolutely no growth at 300 or 500 parts per million with cassia, with cinnamon bark. And then you have other oils like palmarosa and thyme and geraniol that at 500 parts per million really suppress growth very significantly, and at 300 a little bit, so still quite a significant reduction in growth, but not quite as strong as cassia and cinnamon bark. So for the FV, and I'll show you some actual data here in a second, the most promising oils have been cassia, cinnamon bark, and then these other three. All of them have more than 80% growth inhibition in these Petri dish assays. And so here's some data. This is just shows only 8 of the oils we tested. So I'm not showing you all the data because it's too much to show in this one class. But basically, you have the essential oils on the x-axis, and on the y-axis the radial growth. So we measure how much the fungus grows, the radius of the fungus in those colonies. The controls are just without the oil. And then we have the 2 concentrations, the 300 part per million, 500 part per million. And I highlighted in red the oils that you can see have the most suppression. As you can see, most of them suppressed growth at some level, except the orange oil in this batch of oils. But really the most effective were cinnamon bark, geraniol, palmarosa, and thyme, especially at 500 part per million, but also about a 50% reduction at 300 parts per million. So again, very low concentrations of the product needed to suppress the growth. So that was our main work on Physarum verrucoliforme, but again, we're trying these other pathogens to see if there's an oil that maybe is effective against a variety of organisms. So these are the results on Macrophyllomina phaseolina. Macrophyllomina has an interesting characteristic. It's a pigmented fungus, so it forms microsclerotia, and they get melanized, they get very dark. That's why the colonies look so dark. And so it's a bit of a much harder fungus to control, and in fact there's no fungicides that are effective against Macrophyllomina, and there's also no host resistance. So even though most of the oils had no effect on Macrophyllomina, we were pretty excited that we see that cassia, again, completely inhibited growth of Macrophyllomina, and cinnamon bark, not as strong, but a little bit less pigmentation at the 500 parts per million, and manuka oil, which is a different kind of oil, also showing a significant reduction in the growth rate of this fungus. So the implications of this we'll have to explore further, but even a reduction in the growth rate might be something that could slow down infection and potentially help manage the disease. But Macrophyllomina, these 3 oils inhibited growth by more than 50%, and the pathogen is mostly resistant to most of the other oils. As you can see here, this again is the data set. You have the control on your left, and then all the different essential oils, and everything grew perfectly fine. It's really not much affected except, again, the cassia and the manuka oil, particularly cassia. So exciting to find at least a couple of oils that have some potential. And one thing we're planning to do with these 2 oils is test them at higher concentrations, because again, we're testing 300 and 500 parts per million because that was effective with lemongrass with Physarum verrucoliforme, but there's no reason that we couldn't try 500, 800 parts per million or even higher, and some of the other oils may be effective at higher dose. So that's something we want to explore going forward. Okay, in contrast to Macrophyllomina, that was so very resistant, the results on Pythium sylvaticum are quite the opposite. So Pythium is a bit of a wimpy pathogen in terms of growing in presence of essential oils. As you can see, this is just a selection of plates. There were more that looked like this. The control grew extremely well, and in cassia, cinnamon bark, citronella, clove, geraniol, lemongrass, oregano, palmarosa, and a few others, there was absolutely no growth. So very sensitive pathogen to the oils at 300 parts per million and 500. The only oils that had not much impact on Pythium were caraway, manuka, orange, patchouli, and tea tree. So a huge selection here of oils to choose from. So this was the most susceptible pathogen we tested. And as you can imagine, the graph looked like there's no growth in everything except a few of them. The other pathogen, the fourth pathogen, is Sclerotinia sclerotiorum, the white mold pathogen. And here again, we had a good selection, and you can see there's a consistency here. Cassia, cinnamon bark, lemongrass, manuka. There's a few of these oils that are appearing as being effective against the range of pathogens we're testing. And so you can see here with cinnamon bark, 300 parts per million didn't do much, but at 500 parts per million, there was no growth. Same with lemongrass, and then manuka and cassia oils inhibiting growth, but just not quite as effective. But these four oils, more than 80% growth inhibition with the white mold. So I'm not showing you data on here, but again, we have all of these pathogens tested with the 16 oils. Here, I know there's a lot of numbers here, but mostly I just wanted to show you the approach we're taking. So now that we have these four pathogens and the set of 16 oils tested, we can create what we're calling a heat map, where we're considering, okay, compared to the control, what was the percent inhibition of the growth of these pathogens in presence of each of the oils, with the goal that we can find, for example, if you look at the first row, cinnamon bark, there was 100% inhibition for Fusarium falciforme on average over 2 runs and multiple replications. Macrophyllomina only grew about 50% inhibition. Pythium didn't grow at all. Sclerotinia didn't grow at all. So cinnamon bark here standing up as one of the top candidates, and also cassia, 100% including for macrophyllomina, and then 96% inhibition for sclerotinia. So when we look at this heat map, we can see which oils are effective across multiple pathogens, but we could also see something like the manuka oil with the macrophyllomina that wasn't very good with Fusarium falciforme or Pythium, but was quite effective with macrophyllomina and sclerotinia. So we can also start pulling out oils that may be used to target a particular pathogen as opposed to a set of them. Okay, another thing I just want to mention before I move on is that we are also testing for fungicidal versus fungistatic effect of these oils. So what I'm looking for is, are these oils just suppressing growth while the pathogen is in contact with them, or does it actually kill the fungus? So what we do is on those plates that have no growth, we take that little plug and put it on a plate with no oil and see if they regrow. And several of these oils are actually fungicidal, so they prevent the growth of the fungus, so they kill the fungus rather than just inhibit it temporarily. So we're going to look and explore that a little bit further. Okay, so one question that comes up a lot when we talk about plant essential oils is phytotoxicity. So if these oils are so powerful against pathogens, won't they affect the plant itself? And it's a very important question that we are also looking into, because if we're going to be able to use these, we have to make sure that they're safe on the plant. And so we are doing what we're calling phytotoxicity tests, and we're doing this in 2 ways. One, we're using a plate assay. So we amend the agar with different oils at different concentrations, and then we surface sterilize seeds and germinate them basically directly on those agar plates to see if there's any effect on germination, and also if there's effect on the length of the root if they do germinate, so any slowing down of the root germination. And this here is just to kind of show you that, in fact, phytotoxicity is something we do have to consider, and we're going to have to address in the future. The top row of slides are for day 10, so 10 days after we plated the seeds. The C1, C2 are controls. One is just a regular control. The other control has the tween because we need that emulsifier. And you can see the seeds germinated very well. You have nice roots and radicals growing there. And when you use lemongrass at 500 parts per million and also 0.5, which is a pretty high concentration of the oil, the seeds didn't germinate or germinated poorly. If we waited a few more days, day 14, so the bottom row, you can see that the lemongrass at 500 parts per million, the seeds are now germinating. So it didn't kill the seeds at that concentration, but it's definitely slowing down germination, and so delaying it, and then also inhibiting at higher concentrations. We've done several assays with this. We noticed that there's a dose dependency, so it's not like it's always phytotoxic. If you reduce the dose, the seeds will start germinating normally. And the other thing about this assay that we concluded, it's a bit not quite representative of what we want to do with these oils, which is basically apply them as a seed treatment. That will be a very thin layer of the essential oil over the seed. So we have moved from these plate assays to different types of assays to be more realistic in terms of how much exposure the seeds will have to the oil. So these are the plate assays, definitely highlighted the need to look at phytotoxicity. The other assay we've been working and developing is using germination paper. So we call them roll towel assays or germination paper assays. So we have that brown strip is just germination paper. We prepare the different oils in water with a little bit of water agar, just so it's a little thicker of a substance so it sticks to the seed. And so we have this 1% water agar with the Tween 80 emulsifier, and then we apply the oils at concentrations of 1% and 5%. And you'll notice that these concentrations are quite a bit higher than what we did in the plates. And that's because these concentrations are more similar to what our colleagues are doing in terms of delivery method. So we wanted to up the concentration also so there's enough oil around the seed since it's much less contact than on the plate. So we did up the concentration here. We apply these emulsions to the seeds, coat them, let them dry a little bit, and then we roll them in these paper towels and put them in a tub with water and let them germinate. And you see the little seedlings starting to pop out of those roll towels. So then we measure germination percentage and we measure the root length. And again, here, I'm just showing you one example here with the geraniol, just one of the oils we tested. On the top left, you have the tween control. And you can see we don't have 100% germination. It's about 80%, a little over 80%. So some seeds are just naturally not going to germinate, but pretty good germination after a couple of weeks. And with geraniol at 1%, applied as a seed treatment, we see some, maybe a little less germination. The roots may be a little slightly smaller, but then with 5% geraniol, you start really noticing a difference. And if you look at the graph on the bottom there, you can see 1% geraniol, it's just slightly under 80% germination, but 5% geraniol, closer to a little bit over 60. We are going to continue to test these. We're actually also looking into an assay, phytotoxicity assay in a in a pot with soil. So treating the seeds, plate them in soil, and seeing how that interaction affects phytotoxicity. And we want to do this before we start doing pathogen assays, because if one of those most powerful oils is extremely phytotoxic, it's not going to work for us. So we want to test inhibitory effect against the pathogens, phytotoxicity on the seed, and then, as I'll talk about in a minute, we're going to look at delivery methods to then look at effectiveness against the pathogens in terms of disease development. So clearly, with essential oils, there's opportunities and challenges, right? Like with everything else. So we have these oils. Opportunities are they have very strong antimicrobial activity, especially on these fungi that we tested. They're very effective at very low levels, at parts per million concentrations. They are biodegradable, so that's a benefit compared to synthetic fungicides, and they're generally regarded as safe. So these are all great things, but we do have the challenges of, one, volatility. So these oils will eventually volatilize, that we smell them, right? And so they disappear and will stop having effectiveness. So we need to look at that. How long will they last? The phytotoxicity that I just talked about, and the fact that they're biodegradable is a plus, but also a negative, right? Because if they're very quickly biodegradable, then how long will their activity last? So the question we're trying to answer in my lab now going forward is, can we maintain the opportunities while addressing the challenges, and what are some approaches we could use for this? And I'm sure there's many different ways to do this. In my lab, what we're trying to look into is the use of nanotechnology to see if we can maintain these advantages while addressing and reducing the challenges. So before I go into nanotechnology, I'm going to just give you a little bit of background on nanotechnology and nanoencapsulation. So we are collaborating with a couple of colleagues here at ISU with the idea that we will try and encapsulate the essential oils that are most effective from our screens into nanoparticles. And so nanoencapsulation is basically enclosing, creating sort of a shell around the active ingredient, in our case, they're the essential oils, using particles that are at the nanoscale. A nanoscale is basically 10 to the power of -9 of a meter, so a billionth of a meter, so very, very small particles or droplets. And they have many different applications with medical uses and pesticides and many other things. But the benefits of nanoencapsulating an active ingredient is that you can increase stability of that active ingredient. You can have controlled release, because whether they're in a capsule or a nanosphere or nanoemulsion, these active ingredients are more slowly released into their target. And also because they're so small, they have high absorption rates into the plant, into the cells, and so they reach the target effectively because they're so small. And so we are looking into the use of this nanoencapsulation to see if we can solve some of the challenges we're seeing with the plant essential oils. So I want to introduce two of my collaborators here on campus. One is Dr. Carmen Gomez. She's in mechanical engineering, and also Dr. Lingling Liu from Ag and Biosystem Engineering. And both of them are experts in nanotechnology, and we are working together with different projects to see if they create nano materials that are bio-based. So Dr. Lingling works on cellulose that she prepares from corn stover or soybean residue. So it's a use of these residues to create a nanomaterial that's natural. Dr. Carmen Gomez uses several compounds, but in the case of our project, she's using alginate from algae and zein from corn. So these are, again, products that they make nanoemulsion, and then we will emulsify the oils in those nanoemulsions with the hope that this will benefit enhance the ability to use the essential oils as treatments, as seed treatments, or even as foliar treatments, because they will reduce the volatility, because we encapsulate the oils, and so there's slower release of the active compounds, and also increase the effectiveness of these essential oils, because again, they're small and they will be able to more easily be absorbed into the pathogen, killing it, or into the plant and enhancing defense mechanisms. And also, one of the key roles is to see if we can reduce phytotoxicity, because if they are enclosed in these nano emulsions, the concentration is diluted, right? It's releasing more slowly, and we may be able to protect the plants from the phytotoxic effects. So this is work that they're doing with the oils that we have screened, and then we will test the formulation that we'll come up with in terms of disease suppression. So just to give you a little bit of a visual what these nano compounds look like. This is some images from Dr. Liu lab. So she uses soybean residue. She uses the stover. She breaks it up into small pieces and then washes and grinds it, and then is able to, through some biochemical methods, extract the cellulose. So you can see that image on the right is basically like a gel, and it's at nanoparticle size, and then that gel is mixed in with the essential oils, and so it causes an emulsion of those oils. And so the different compounds they're testing will have similar properties. They're characterizing these properties for stability and all these other characteristics that will be important to develop an effective delivery system for the essential oils. Nanocellulose in this case, but also the alginate and the zein and other bioproducts that we're testing are non-toxic, they're biodegradable, and they have already been shown to be effective nanoencapsulation agents. So that's very promising on that side. Okay, so just wrapping it up, we have some strong evidence that essential oils have strong antifungal activity at very low doses against 4 of the pathogens that are very important in soybean. We do have challenges with volatility, degradability, and phytotoxicity, and so we're in my group, we're trying to address those challenges by using nanoencapsulation to develop an effective delivery system that not only enhances or at least maintains the effectiveness of the oils against the pathogens, but also reduces the phytotoxicity and the volatility, so that's more of a durable effect. Looking forward, if we are able to develop an effective delivery system, what we envision is that we will be able to use these nano-encapsulated products as a novel integrated pest management tool that can be added to the other tools that are used by growers. Maybe for example, organic growers, this could be an alternative to synthetic pesticides. So you could just apply these treatments instead of synthetic pesticides. But it's also possible that we could, for example, be able to reduce some of the phytotoxicity effects of some of the synthetic pesticides by reducing that dose and complementing that treatment with some of these plant essential oils. So in my lab, our goal is to develop seed treatments that could be applied as a coating to the seeds. But I think that the technology that we're developing and the information we're going to gather through this collaboration would be useful also for foliar applications, and then eventually for other pathogens and other crops. But we still have a lot of work to do. And so before I end, I'd like to acknowledge the people that have contributed the most to the research I presented here today. Most of the data, especially the data on Fusarium virguliforme, was collected by my PhD student, Alexandra Starkey, who is doing her dissertation on this topic, and then also by two undergraduates, Diana Mulder and Brady Clausen, that have been collecting data on the other pathogens, and then Vijita Silva, who is my research associate, who is kind of helping with all the projects. And also like to thank my collaborators, Dr. Ling Liu, Dr. Carmen Gomez, Victor Wanderley, who's a student in Dr. Gomez's lab, Dr. Joel Coates, who's an entomologist here at ISU, who has more than 30 years of experience working with plant essential oils as an insecticide, and so we've learned a lot from him, and he's provided a lot of resources to help us get started. And finally, I wanted to thank the Iowa Soybean Association and the United Soybean Board, both of which have funded the work we've been conducting, and we are excited to continue to work on these projects and come back later, different time, to give you an update. For Iowa State University, this is Leonor Leandro.